able to form their own organic molecules for food. Plants are autotrophic. Heterotrophs are organisms that must consume other organic compounds for food, and that means consuming other organisms. Heterotrophs are consumers. For example, humans are able to obtain nutrients by consuming other organisms or the products of other organisms. Right here we're looking at a trophic pyramid. At the bottom we have producer.
我想吃炒饭，我等会儿自己做。对。
Then we have our primary consumers, followed by our secondary consumers and our tertiary consumers. Ali? Level in a trophic pyramid, we know that energy is lost. Usually it is heat, but up to 90% of energy is lost every time you go up a level in an energy pyramid. We know that it's more efficient to consume lower on the pyramid than it is to be a tertiary or quaternary consumer. In primary organisms, right here we're looking at a trophic pyramid.
These notes are for a living environment. Hi.
At the bottom we have producers, then we have our primary consumers, followed by our secondary consumers and our tertiary consumers. At each level in a trophic pyramid, we know that energy is lost. Usually is heat, but up to 90% of energy is lost every time you go up a level in an energy pyramid. We know that it's more efficient to consume lower on the pyramid than it is to be a tertiary or quaternary consumer. In one ecosystem, we have lots of biotic, living, or abiotic factors. And an ecosystem is shaped by the interactions between these non-living and living factors. In every single environment, there is competition between different organisms.
I got the cup while we were on vacation. I'm studying.
In place that keep populations and ecosystems relatively stable over hundreds or thousands of years between the than it is to be a tertiary or
what grade? I'm in 8th grade.
Quaternary consumer. In one ecosystem, we have lots of biotic, living, or abiotic factors. And an ecosystem is shaped by
College major? Not exactly, but I plan on doing something in aviation, I guess. How old am I? I'm 13. I'm not writing an essay. Oh yeah, I do have Regents. I have Regents. I'm taking 2 this year.
The interactions between these non-living and living factors. In every single environment, there is competition between different organisms and interactions between different populations.
I'm taking the living environment tomorrow. I'm not doing homework. I'm trying to study for the living environment.
Within one ecosystem or environment, there are natural checks in place that keep populations and ecosystems relatively stable over hundreds or thousands of years. So sometimes we might see certain individuals that have what looks like unlimited or exponential growth. Most of the time, most populations will reach what we call as a carrying capacity.
Yeah, I'm taking the science Regents. I'm from the United States. Show me what you're reading. Yeah, this. It's backwards, but... Oh. I don't know. You got pretty lucky. Yeah, I'm taking my second science Regents this year. Age, I'm 13. My reading is so bad. Okay, well, deal with it. I'm reading this. Do I write a lot? Not really, not typically. I don't have business. Well, I'm studying a lot because I don't want to forget stuff for tomorrow, and I'm like missing most of the lessons, so, and I don't have notes from some of them, so I'm doing it now.
Capacity within a particular environment. Because there are limited resources within a particular environment, like food availability, water, space, at some point those organisms are going to reach the maximum population that that ecosystem or environment is able to sustain. And even though that may fluctuate over time, a population may be relatively stable once it hits its carrying capacity. If we see a sharp decline like this, it's probably because there was an introduction of a disease, or a new predator, or an invasive species. Let's bring humans back into this equation. Humans are pretty complex. We're made of molecules, and groups of those compose our cells. Groups of cells compose different tissues in our body, and different groups of tissues can make up our organs. Our organ systems are several different organs working together. For example, our digestive system, our circulatory system, our respiratory system, our excretory system.
I'll just get another pen. Why are you wishing the worst on me, man?
Functions that are essential to our survival. We have different specialized cells for different functions in the body, and these cells can work together to perform specialized functions. One of our characteristics of life is that
English. I don't know Spanish. I do know Dora. Yeah.
all organisms maintain homeostasis.
my science labs. Yeah, I did all of them. I finished the science hours a while ago. Where am I from? The United States. Or like if you're speaking of ethnicity, China. Dude, I don't go by car. New York.
Or stable internal balance. Organisms have to maintain homeostasis in order to survive. Often, the result of not maintaining homeostasis is often disease or death. As an example, humans sweat as our body temperature rises. Usually, our brain is going to send signals to our blood vessels to dilate, which is going to help us lose heat in the environment. Then our sweat glands
Dude, what did I do to you? Why are you hating?
parts of the body are
if you
also
have a problem with me, just like leave.
Cooling, our body will start to cool down. All of this is part of homeostasis. We also can shiver in order to maintain homeostasis. And organisms that aren't able to regulate their temperature internally will often move to a warmer location or a cooler location in order to help maintain that stable internal environment. These organisms, like lizards, are ectotherms. Remember, if there's any disruption in a human body system, there might be a disruption in homeostasis. When we look closer at our cells, we know that these are specialized structures with organelles inside of them. And
Okay, doesn't matter what I look like. If you have a problem, just leave. I'm not forcing you to stay.
organelles are specialized structures with specific functions within the structures with in homeostasis. When we look closer at our cells, we know that these are specialized structures with organelles inside of them. And organelles are specialized structures with specific functions within.
Palestine? Dude. I don't know who you are, but for some reason you're hating. Once again, if you have a problem, please just leave. I'm not forcing you to stay here. I'm trying to like study. I'm trying to like chat with like whoever's here. But I'm not going to force you to stay. If you have a problem, just leave. Please, God.
Then the cell. We see the mitochondria here. We see these dots as representing ribosomes. And of course the nucleus in the center of the cell with our DNA or our genetic information. We see a vacuole for storage here, and we see our cell membrane which surrounds the cell. These organisms, like lizards, are ectotherms. Remember, if we look at our cell, we know that these are specialized structures with organelles inside of them. And organelles are specialized structures with specific functions within the cell. we see the mitochondria here. We see these dots as representing ribosomes within the
Shout out to Alex. I don't have to wear a uniform. What am I gonna do? Just wear something else, man. Honestly. Christina. Dude, Daniel. I don't care what you think I look like. If you have a problem, just leave. Honestly. What is wrong with you? Do something with your life. No, I don't need to wear any uniform. I could literally just wear something else, man.
cell. We see the mitochondria here. We see these dots as representing ribosomes, and of course the nucleus.
Wear a uniform now? Dude, I'm at home. Why do I need to wear a uniform? I live in the United States. I'm not required to wear a uniform. Can you guess how many brothers I have? 2. I'm taking a guess. What am I writing for tomorrow? I'm not writing for anything. I'm just studying, taking notes. Do I have good handwriting? Not really. It's just enough for me to read it. Wear a hat? I don't have a hat. Living environment regions? In which class do you read? English class, ELA, huh? Why would I give you a shout out? Dude, you were hating just now. Which class do you read? Like ELA. Whose student am I? What am I studying for? I'm studying for living environments. Living environment, science. Parents? My parents are kind of strict. It's not like they are obsessively.
Nucleus in the center of the cell with our DNA or
Deal with it. Yeah, I think I banned Rhonda. What form of science? Living environment. Do I get good grades? I mean, relatively good grades. It's not like I'm failing or anything. Am I in high school? No. I'm in middle school. I'm in 8th grade.
for our genetic information. We see a vacuole for storage here, and we see our cell membrane, which surrounds the cell and allows for molecule.
Super seniors? Yeah. Do I know how to cook? Yeah, I do. I have cooking streams sometimes. Am I in 5th grade? No, Daniel. Do I have siblings? I have, a younger brother. What do I want to be when I'm older? I mean, I'm hoping pilot. It has its advantages. Do I have a boyfriend? No. No, I'm not in 4th grade either, Daniel. God. Hello. Do I like anyone? No. I don't know if you guys can see when I time out someone.
To move in and out. Our plant cell over here also has a cell wall and
Does someone propose to be in a relationship? I mean, multiple people, but I'm not, like, trying to be in a relationship, so...
well. Through diffusion, in and out. Our plant cell over here also has a cell wall and a chloroplast.
What's my favorite hobby? I like to read, sleep. Yeah, that's about it. It's not like I don't like relationships. I would just like to stay out of them for now. And like honestly, the guys at my school aren't exactly like really relationship worthy, so reading I like dystopian books and yeah, romance. Dude, Saurabh is also switching up, honestly. No, I don't have any crush. Do you have any book recommendations? Okay, so I have a shelf of books behind me. So for romance, the Twisted series was good. Something about it really hits me the wrong way though, but it was actually really good. Then if you want like romance that's like not fully like circled around romance, but there's still romance in it, I recommend the Natural series and A Good Girl's Guide to Murder. It's both like mystery series with like a little romance twist.
As well, through diffusion through our cell membrane or through special transport proteins. We can get the molecules we need to break down either for energy or other important processes within our bodies. Generally, larger organic molecules need to be broken down before they're taken in by our cells.
Books? Yeah, the books I just told you about, they're actually like really good series. Yeah. There's also dystopian novels that I haven't read. Like, Divergent, I heard that's really good, so I have it, but I haven't started reading it.
Cells, but once they enter the cell, our cells can use them as the building block for energy, or through here also has a cell wall and a chloroplast as well. Through diffusion through our cell membranes or through special transport proteins.
Oh, Cruel Prince? Yeah, I've heard of Cruel Prince. I have it on my lists, but I don't have it yet.
Get the molecules we need to break down either for energy or other important processes within our bodies. Generally, larger organic molecules need to be broken down before they're taken in by our cells, but once they enter the cell, our cells can use them as the building blocks for other important molecules within our bodies. Like once they enter the cell, our cells can use them as the need to be broken down for energy or other important processes within our bodies. Generally, larger organic molecules
Do I have any pets? I have fish.
Need to be broken down before they're taken in by our cells, but once they enter the cell, our cells can use them as the building blocks for other important molecules within our bodies. Like I showed you here, sometimes the mitochondria might be simplified into an oval with a little squiggle line. Sometimes it may look a little bit more complex where you can see all of those inner folds or cristae within the mitochondria. The chloroplast as well can be a little...
What animes do I watch? I've watched a lot. I have a little list in here. Yeah, this is all, like, this is, like, just some of the animes I watched. Another page. And there's more. Fish. I didn't name my fish. I just kind of have them. I have, like, I think 5.
More complex than this drawing here, where you can see these stacks of thylakoids where important processes of photosynthesis take place. Remember, our mitochondria within our cells is used for energy production. It's the site of aerobic cellular respiration within our bodies. A vacuole is used for storage. Ribosomes are there to make proteins. The nucleus is the storage site of DNA in eukaryotic organisms. And our plasma membrane is a semi-permeable barrier that is made of phospholipids. Cell wall is there for structure and support, and the chloroplast is a location of photosynthesis. A few main differences between eukaryotic and prokaryotic cells is that eukaryotic cells typically are more complex, have membrane-bound organelles, are larger, and have their DNA enclosed within a nucleus. Prokaryotic cells have very few organelles. In fact, they just generally have ribosomes and some other internal structures. Cells are larger. This is a location of photosynthesis. A few main differences between eukaryotic and prokaryotic cells. That eukaryotic cells typically are more complex, have membrane-bound organelles, are larger, and have their DNA enclosed within a nucleus. Prokaryotic cells have very few organelles. In fact, they just generally have ribosomes and some other internal structures, but no membrane. Bound internal organelles. They'll have a cell membrane, they'll have ribosomes, and they'll have their DNA, but it won't be enclosed in a nucleus. Sometimes they may have cilia or a flagellum. Prokaryotic cells are, remember, cells like bacteria or archaea. They came first in our history of life on Earth. Eukaryotic cells are more complex, and they include cells like animals and plant cells. Cells can also communicate with other cells or other parts of the body through special signaling pathways. For example, this protein embedded in the cell membrane can receive signals from other parts of the body. These are receptor molecules, and they play an important role in interactions between different cells. pathways. For example, this protein embedded in the cell membrane can receive signals from other parts of the body. These are receptor molecules, and they play an important role in interactions between different cells. For example, hormones that are released by different glands in the body will then be sent to different cells within the body for a particular reaction. The nervous system will produce signaling molecules like neurotransmitters that will allow us to have different effects within the body as well. But if our signaling pathways are disrupted or our receptor molecules are blocked, for example, our cell communication can be disrupted and the cell or the organism receptor molecules are effects within the body as well. But if our signaling pathways are disrupted or our receptor molecules are blocked, for example, our cell communication can be disrupted and the cell or the organism itself and their stability could be affected. For example, when we consume caffeine, caffeine actually blocks the signaling molecule for adenosine, which is going to make us sleepy. When we don't receive those special signals anymore, our body will feel less sleepy and will feel less tired. Single-celled organisms have specialized structures and functions as well that can help them perform tasks similar to that of multicellular organism. For example, a pseudopod within a particular single-cellular organism can help organisms move. Remember, we get a lot of our traits from our genes, which are inherited, but the expression of our genes can be modified by the environment. And we can see this in humans when we see separation of identical twins.
Your child needs these 3 key factors to get into an Ivy League or top 25 university. And it's not having a good GPA or SAT score. Let me explain. 90% of students with a 4.0 plus GPAs get rejected from universities like Harvard, Stanford, and other top 10 universities. Top universities made SAT and ACT submissions optional. And with over 100,000 plus applicants a year, as a parent, you know your child needs to stand out. Top universities like Harvard, Stanford, Princeton, and others admit students who will have a successful career after graduation. This is what gives a university its prestige. Universities are businesses at the end of the day. And if they have great alumni who become successful, it's the best form of marketing for the university. And we know this because we've guided thousands of students in over 40 states to get into the top 25 universities. And we know that there are 3 factors in the admission criteria that universities are looking for. And that's why we have a free webinar where we go over these 3 main factors that prevent 6th and 12th graders from getting into the top colleges. And we covered the important
Twins. If identical twin girls are in humans when we see separation of identical twins. If identical twin girls are separated at birth and grow up in 2 different environments in 2 different households, we might see that after 20 years, the different girls have different heights and weight. This is because each twin was provided a different diet, had different physical activities, and was exposed to different environmental factors throughout their lives. So genes don't determine everything, and the environment can have a huge influence on your genes as well. Most of the traits that we have are actually influenced by multiple different genes at once. And in our own nucleus, we have thousands of different genes. When we say genes, this is just a section of DNA that's going to code for a specific protein. DNA, remember, is a nucleic acid. It's made of nucleotides, and one nucleotide is composed of a phosphate, a sugar, and then that base. A And the order of these bases within a DNA is going to determine what proteins that DNA will code for, and those proteins will give us the traits that we have. Now, all of your cells in your body have the same DNA, however, different parts of that DNA are turned on and off in different cells. So for example, your muscle cells will contain the same DNA as your skin cells, but they won't have all of the DNA working all at once. It's like a cookbook. No one chef is going to cook all of the recipes at once. They're going to pull out a few different recipes at a time and cook those for dinner. Think of the DNA like the cookbook of your cells. All of your cells have the same cookbook, but we're only cooking a few recipes in each different cell. When we zoom out and we look at multiple nucleotides put together, we see that each base, either A, T, G, or C, is going to connect with a complementary base on the other side. DNA is a twisted ladder or a double helix, and each base will match up with its complement connected by a hydrogen bond. Our base pairs are adenine and thymine, so A always pairs with T, and guanine and cytosine, G always pairs with C. You want to make sure you remember this and try to think of a way that it's easy to stick in your head. For example, you can think of apple tree, good cookie. I've also heard all teachers can go. Whatever sticks in your mind, help you remember your base pairing rules. This is something that'll be important for your biology studies. So for example, if you need to write the complementary or the matching base pair sequence of this strand of DNA, you would remember that T pairs with etc. and instructions for proteins in a strand of complementary DNA. Now remember, DNA is going to hold the instructions for proteins in a cell. That DNA out of the nucleus, protein synthesis, more specifically built in the ribosome. The process of making protein is called protein synthesis. More specifically, it's the process by which DNA is translated and transcribed into proteins. So our DNA code is transcribed into mRNA. Our mRNA is then translated into a chain of amino acids to form a protein. Protein molecules are pretty long. They're made of 20 different types of amino acids. And what's crazy is that all organisms on Earth share the same universal genetic code. Remember that the sequence of amino acids within a protein is going to determine the structure and then the function of that protein. If we change the order of amino acids, we could change the shape of the protein and we could change how it works. Now, we use all of our knowledge of DNA within genetic engineering and biotechnology. For thousands of years, humans have been doing artificial selection and selective breeding, where we have new varieties of cultivated plants and domestic animals because of the ones that we select to breed and pass on their traits. We can get a little bit more specific, manipulating genes in other organisms, though, through genetic engineering. One of the tools for that is gel electrophoresis, also known as a DNA fingerprint, which is used for forensic analysis, paternity testing, or studying evolutionary relationships. Transgenic organisms are organisms like plants, animals, bacteria that are created for agriculture and industry using genes from multiple organisms. For example, we can produce BT corn, which has specialized genes that can fight off different insects. We can also modify bacteria to help us clean up oil spills. Taking a closer look at gel electrophoresis, we know that DNA is negatively charged, so what we can do is take our DNA, put it into a specialized gel, and run an electrical current through it. At the top, there's going to be a negative charge. At the bottom, there'll be a positive charge. Because DNA is negatively charged, it's going to go towards that positive end of the gel, and we'll start to see the bands of the DNA separated because of enzymes that have cut up the DNA in special places. Now here, we see evidence from a crime scene, and we're going to look at it compared to the DNA from different suspects we find. We look to see where the bands have matched up, and then when we see similar bands, we can confirm that the suspect, and here with suspect 3, is the one that most closely matches the DNA found at the crime scene. Now we can use similar gel electrophoresis techniques or